So thank you very much. This is the second speaker will be Klaus Wimmers. So Klaus, if you can come. And Klaus Wimmers is a farm animal scientist from the Institute of Genome Biology in Dürmersdorf, Germany. He dedicated his work to animal genetics and developed a specific interest on epigenetic mechanisms and their implication in animal breeding. Please, you have the floor for 15 minutes.
Thank you, Mr. President, for the kind introduction. Can you hear me? No, it's not. Okay. Now it's on. Okay. Yeah. Dear ladies and gentlemen, I also like to thank the organizers and the committee members for inviting me for this talk. And yeah, we are dealing with epigenetics. So I mean now, after the genome sequences are known, also from our farm animals, we are more interested to learn about the function of the genome, and so epigenetics becomes into focus. And also, if you think about the fact that from a single cell, a zygote with a certain genetic information, a complex organisms develops, then it becomes clear that on top of the hardware of the genetic information, we need a kind of additional software program that points out how to use the whole information. So that means in the genome, in the nuclei, we have at least two kinds of information: the genetic information with a general instruction how to manufacture proteins, which determines what can happen, the whole potential. This is coded in the DNA sequence. And we have epigenetic information. That is, in addition, instruction on how and when to use this information. And we are often talking about epigenetic marks. So, in animal farming and animal breeding, we are dealing with prediction. We are trying to link the genetic information with the phenotype, and in current days, we have done many efforts to look along the genotype-phenotype map and to try to explain how the DNA information comes up to a final organismal phenotype. And so we have now tools to look at the transcriptome, proteome, metabolome, and so on. And with epigenetics, there's a new level of variation that we can study now. And the interesting thing with this is that epigenetics, as mentioned in the talk before, really links between the environment and the genotype. Via epigenetic mechanisms, the environment can generate certain marks on the genome that have impact on the gene expression. I mentioned here the mRNA or other non-coding RNA. This is sometimes covered with the term epigenetics, sometimes not, since there are still some different definitions around. So originally, Conrad Weddington in 1942 studied epigenesis, and he was dealing with the fate of the cell, so how to come from a zygote to an organism. So it's the science on all these developmental facts. Later on, Holliday defined it as the temporal and spatial control of genes acting during development of complex organisms. And I think in current time, maybe the best definition comes from the NIH roadmap epigenomics project, and they say epigenetics is the study of mitotically or meiotically heritable changes in gene expression that occur without changes in the DNA sequence, and of stable long-term alterations of the transcriptional potential of a cell that are not necessarily heritable. So, what is the molecular basics of epigenetic marks? There are there's DNA methylation and there are histone modifications. These are the main molecular equivalents of epigenetic marks. I will just explain a bit of it because I think that we have a bright variety of disciplines in the room. So, histone modifications actually mainly occur at the tail of the H3 histone, where certain amino acids became modified by adenylation, methylation, and so on. And these changes have an impact on the way the DNA is packed by the histones and whether it is accessible for transcription or not. And there is currently what we call the histone code theory. That means that the different changes that may occur that you see at the bottom of the slide have certain meanings. And this is not fully understood, but it is already focused on this. And you may be aware of the FANG initiative for functional annotation of animal genomes. And as you see here, in this initiative, it was decided to address 4 specific histone modifications that are functionally quite well known and where antibodies are available to detect them. The other mechanism of epigenetic, which is much more studied, is the DNA methylation. DNA methylation occurs at the cytosines and where a methyl group is introduced, and this causes usually a stable inactivation of gene expression. It allows a long-lasting gene expression control. It is involved in imprinting, and there are also mechanisms of acute gene regulation. How does this occur? There are certain so-called DNA methylases, and these DNA methylases can put in de novo, so new methyl sites, as you see on the right edge, and during replication there is a maintenance DNA methyltransferase active that just adds the missing methyl groups on the new strand. And there are also demethylase enzymes that can erase these marks. So in the genome we have about 1% of the CpG denucleotides where DNA methylation occurs, and this is a lower percentage than should be expected statistically. So these CpG sites are usually highly methylated. You see about 60 to 80% are methylated, and many of these CpGs are arranged in so-called CpG islands. These are regions of about 200 base pairs, which are only at 60% CpGs, and these are usually located in promoter regions and are usually hypomethylated, so allow activity of a gene. And CpGs outside of these CpG islands are usually strongly methylated, and DNA methylation has a functional, also an evolutionary role, and this is in maintaining genomic stability, especially in long-term inactivation, for example, of repeats and retrotransposons. And I want to mention here that there is a reaction that is deamination of cytosine, which results in uracil. When this happens, this will be repaired. But when methylcytosine is deaminated, we are getting thymine. So we are getting another base. We are generating a mutation that might be an important point that happened during evolution, and maybe still. So what happens at these methylated sites? So they cause a direct blocking of transcription factor binding sites, or they recruit methyl binding proteins, and these methyl binding proteins can in a further step also recruit enzymes that introduce histone changes. So some epigenetic phenomena that I would like to mention. you may have of X chromosome inactivation and of course of genomic imprinting. So X chromosome inactivations happens actually by the activity of a certain locus on an X chromosome which is activated, which codes for a long non-coding RNA, and this long non-coding RNA then covers the X chromosome in cis. So the chromosome that is expressing this gene becomes inactivated. And the interesting thing with this is that this happens during development of the blastocysts in each cell individually. So that means with regard to this X chromosome inactivation, the organisms are mosaics, and this is the cause, for example, for these color patterns that we observe in cats. So when does all this happen? We are talking about epigenetic reprogramming. So after the zygote has been established, there is immediately a demethylation of the genomes happens, and this happens a bit faster in the maternal genome than in the paternal genome than in the maternal genome. And later on at the blastocyst stage, the methylation status is re-established. And of course, many somatic cells just keep this methylated states, which is inherited with each methylation, with each mitosis. But for the primordial germ cells, there's another phase of demethylation and establishment of a new DNA methyl pattern. And of course, there are, for example, repeats that remain methylated all over the time. And this is an interesting point, there are the so-called imprinted genes. So genes that have some epigenetic marks that are either expressed only from the father or only from the mother. And so that means that in this case, either the maternal or the paternal alleles remain methylated during these processes. And we know this imprinted genes from experiments with where the combination of only paternal or only maternal pronuclei failed to give rise to a living animal. So nowadays we know about some more than 100 imprinted genes in mouse, and you see quite a few number in the farm animals. And these genes are often clustered in certain regions. And I will just show you one interesting example. You might know the IGF-2 locus, which is combined in a highly methylated region. And you see on the maternal allele, the sites in the middle are not methylated, so the CTF can bind. So then the H19 is expressed. And this will make that there is no IGF-2. In the paternal case, these methylation sites are methylated, and so the CTF cannot bind, and then an enhancer becomes active and IGF-2 can be expressed. And interestingly, there is a point mutation in this IGF-2, and this point mutation has the effect that a repressor on the IGF-2 cannot bind to it, so it impacts its activity. And so if you have a paternal A allele, then this is highly expressed, and we have high levels of IGF-2. And this is a nice thing that could be applied in breeding programs. So we could establish a sire line, which is IGF-AA, and sow lines, which have the G allele from the paternal side. And so we have sows with improved polyphycracy and longevity, and we have improved meat and lean content and uniformity in the slaughter pigs. Other implications of epigenetics. So, of course, epigenetics is an additional source of variation, and its knowledge may contribute to the prediction of the phenotype. So, if we do know what impact certain epigenetic marks have, we could consider this in our modeling and genomic estimates. Of course, the epigenetic marks could be used also as biomarkers, so we could regard them as a kind of phenotype that is predictive for the later phenotype of an animal. It's interesting, for example, with regard to behavior, as we heard just before. If we can detect epigenetic marks that affect behavior of the animal early at life, we can use this as information for management, the keeping of the animal. I mentioned that the knowledge will contribute to the refinement of estimates, and for example, if you know an imprinting locus, we could consider them as a functionally hemizygous locus. There's an ongoing debate whether these epigenetics contribute to the missing heritability or missing causality that we observed. And there are two situations. So if we find that epigenetics mutations are stably inherited across generations, then we will also detect them by our current genotyping procedures, since we can expect that they are in linkage disequilibrium with the SNPs. So it doesn't matter. And if these epimutations are not stable, then of course they are not a good tool, not a good selection criterion, and they do not contribute much to missing heritability. But if the epimutation is causal, whether transgenerationally inherited or not, it is important to detect it, because for some traits we won't find any variation on the DNA sequence level that is causal. So the epigenetics have impact on assisted reproductive technologies, and I think the next talk will cover these. And there's an area of fetal programming that we already heard in the talks about the mouse experiments before. So there are epidemiological data like the Dutch hunger winter, for example, and other experiments in model animals that revealed that there are environmental effects during gestation on the phenotype. And so epigenetic mechanisms are the molecular memory that are involved in these processes. There are a lot of experiments done, and we here displayed, made a review. You see that these gestation diets were fed at different times, from before matting until post-weaning, in various experiments. And what is marked in red is what we have done. We have done gestation diets with high and low protein and with high methylating micronutrients. I just want to remind you about what does it mean if we do this feeding along the whole gestation. That means we affect, of course, the mother, which is generation 1. We affect the fetus, which is generation 2, and we affect directly the primordial germ cells, which is generation 3. So if you want to show transgenerational inheritance on the female side, we need to look at generation 4. For the male side, it's generation 2. So we had one experiment together with our colleagues from the nutritional department, where we used high and low protein diets, and we were looking for the impact on the gene expression and epigenetic methylation. And I don't want to go into detail for these experiments. I just want to remind you here what we find is that there is an hierarchical influence of tissue, stage, and diet on the transcription level. That we found that in an overall meta-analysis of all the data we covered, that muscle appears to be less resistant to nutritional modulation than liver. We did not find any gatekeeper pathways or genes, which was hypothesized before, that there are certain pathways that are affected in all cases of nutritional programming, but that seems not to be the case. We found differentially expressed expression of the DNA methyltransferases, and we could detect differential DNA methylation in a number of genes. And this encouraged us to conduct another experiment where we fed a diet which was enriched for methylating micronutrients. So we increased, of course, methionine, but also folate, choline, betaine, vitamin B6, and vitamin B12. And this diet was fed to sows. Yeah. And I wanted to mention that actually the same diet was used by the colleague Martin Braunschweig in Switzerland for feeding boars. And he was able to show that the F2 offspring of these boars differed with respect to back fat. They showed a differential expression in liver and muscle, and they found at least one gene where there was a difference in DNA methylation. So this is the only indication of transgenerational inheritance of such gestational diet effects that we have in farm animals. In our case, we did it just sow and offspring, and we were interested to see what happens on the level of the transcriptomes and to map differentially methylated regions depending on stage, breed, and maternal diet. And so we had a design where we used two breeds to see whether there is an interaction of breeds, that means genetics and epigenetic effects. We had two diets and we focused on two stages here. And we did RNA-seq to look for differential expression and RBBS to look for the metallome. Just to give you an insight here, what you see on the left side is the distribution of different functional regions of the genome. So in the inner, the orange one, this is the promoter and the regulatory structures. So they only have a very small proportion of the whole genome. And if you look at the red side, that is what we detect with RBBF, and you see that here the yellowish, the orange ones, this is a CPGs, these are the promoters, are highly enriched with this technique, and then we can look at the base resolution on the methylation of these GPC-rich sequences. So actually what we found is that of the CPGs that we detected here, 43% were methylated, whereas of the C in the framework of other bases, there were nearly no methylation detectable, and this is what we do expect. So here you see a clustering on the basis of the DNA methylation profile of these animals. You see that the animals are sorted by stage, a little bit by breed, and also by diet. So there is an impact of diet visible. And here, view on a certain site where you can see that we can detect sites that differ with regard to DNA methylation between breeds. So you see DL and Pietrin is different, and also between diets, and especially we see here that in Pietrin we have a strong impact of the diet, but we do not have this in DL, so there are breed differences. We found in total several thousand sites with differential methylation that are associated with stage, breed, and diet. And of course, we can see which genes are close to these differentially methylated regions and look for the functions of these genes. And you see here, these are, for example, For breed-associated differentially methylated regions, we found that the WNT pathway or the epithelial mesenchymal transition pathways were affected. For the factor maternal diet, for the diet, we also find this epithelial mesenchymal transition, VNT, but also adipogenesis, which is interesting because I think the lipid metabolism seems to be the part of the metabolism that is most often affected by these dietary interventions. Interestingly, the methionine metabolism was affected in both breeds and also including the DNA methyltransferases. Okay. We also found, of course, differentially expression in these animals. It was a lipid diet metabolism that was affected with the adults, and you see a number of pathways with the embryos, including the IGF signaling. And just another way to look at the differentially methylated sites, we can determine where there are possible transcription factor binding sites in these regions and measure then, of course, side by side the methylation. And as you see here in this example, at most binding sites for transcription factors, we have here an increased methylation due to the diet. We found significant correlation between RNA, between expression and methylation. The correlations were around -3 to -5, not very high, but significant. And this is an example where you can show this significant link between expression and methylation. And finally, I would like to summarize. So epigenetic marks determine the transcriptional potential of the cell, and they represent an additional level of information to explain the phenotype. Epigenetic marks link environment and genome and represent the molecular equivalent of genotype times environmental interactions. So the knowledge and understanding of these epigenetic marks will contribute to the refinement of estimates in selective breeding. Transgenerational inheritance and contribution to missing heritability are a matter of debate. Epigenetics marker studies will provide biomarkers either for management or for breeding. And I think that in future we will be able to introduce epigenetic marks in a targeted way to produce animals that are suitable for certain environments. And with this, I would like to thank my co-authors, my partners, and you for the attention.
Thank you. Thank you very much, Klaus, for these interesting premises. So you can stay here. We have time for some questions. As for the preceding speaker, you are supposed to go to the microphone if you want to ask a question. If not, I have one. You mentioned that a little bit in your talk. Do you think that really behavior could be a sort of one of the traits which could be influenced by inheritance, by epigenetics, or is it? Do we have any examples? We saw that in mice, maybe, but in farm animals, do we have examples on that? And could we think that, for example, if we raise animals in good welfare conditions, the progeny will also be in better conditions?
We came up with this idea because of the mouse studies mainly. What we just heard before, so there are some more studies around that show that stress has an impact on the epigenome, and that will affect the stress response of the offspring. I'm not aware whether this has been shown in farm animals. I don't think so. And I'm quite convinced that it will be probably possible to show the impact of bad conditions but I doubt that we can show the impact of good conditions. But I could imagine that, for example, that conditions during pregnancy impact on the offspring, and that we could probably look at the epigenome of the offspring very early and predict whether this will be harmful to stress or not.
Okay, thank you. A question? Yes, please. Could you give your name and country first?
Good morning. Thank you for your talk. I'm Karen Goossens from Ilvo, Belgium. I would like to come back on the microRNAs, the small RNAs. So in your introductory slide, you showed that there was interaction between the transcriptome and the genome by microRNAs, but there was not an error from the environment by microRNAs to the genome. Don't you think that microRNAs can have influences from outside, from the environment to the genome or the transcriptome?
Yes, I think so, but this is nothing what will put a physical mark on the genome. You see, it is there are many also mRNAs that are switched on or off due to the environment of the organism, and the same is true for microRNAs.
Yeah, I'm referring to some studies that indicate that in the uterus there is interaction from the maternal environment by exosomes and microRNAs secreted in the exosomes to the embryo and vice versa. So.
Yeah, this is okay. This is not my definition of epigenetics. Epigenetics are for me are those mechanisms where we have these physical changes on the base molecules and on the histones.
Okay, thank you.
Okay, thank you. This will be the last question for this talk. So thank you very much, Klaus.